Second, messenger system. The second messengers are intracellular signaling molecules released by the cell to trigger physiological changes such as proliferation, differentiation, migration, survival, and apoptosis, etc. The secondary messengers are therefore one of the initiating components of intracellular signal transduction cascades. The examples of second messenger molecules include cyclic AMP, cyclic GMP, inositol triphosphate, diacylglycerol, and calcium. The cell releases second messenger molecules in response to exposure to extracellular signaling molecules, that is first messenger system. That means the extracellular signaling molecules like hormones or any chemicals, these are acts as first messenger system, and the cell releases second messenger intracellularly in response to exposure to these extracellular signaling molecules, which are known as second messengers. The first messengers are extracellular factors, often hormones or neurotransmitters, such as epinephrine, growth hormone, serotonin, etc. These first messengers are or may not physically cross the phospholipid bilayer to initiate changes within the cell directly, unlike steroid hormones, which usually do it. This functional limitation necessitates the cell to devise signal transduction mechanisms to transduce first messenger into second messenger so that the extracellular signal may be propagated intracellularly. An important feature of the second messenger signaling system is that the second messengers may be coupled downstream to multi-cycle kinase cascades to greatly amplify the strength of the original first messenger signal. Earl Wilbur Sutherland discovered second messengers, for which he won the 1971 Nobel Prize in Physiology or Medicine. The second messenger system can be synthesized and activated by enzymes. There are three basic types of secondary messenger molecules: hydrophobic molecules, hydrophilic molecules, and gases. Hydrophobic molecules, that means these are water insoluble molecules. Hydrophilic molecules, these are water soluble molecules, and gases like nitrous oxide, carbon monoxide, etc. These intracellular messengers have some properties in common. First, they can be synthesized, released, and broken down again in specific reactions by enzymes or ion channels. Some can be stored in special organelles and quickly released when needed. Their production, release, and destruction can be localized, enabling the cell to limit space and time of signal activity. This is a general schematic of second messenger mechanism. Here it is divided into four phases. At phase 1, agonist activates membrane bound receptor, which subsequently causes phase 2, G protein is activated and produces effector. In phase 3, effector stimulates second messenger system, and in phase 4, second messenger activates intracellular process. This schematic also shows the same in another format. This is a very simple schematics showing the same phases. Here green color showing agonist, which attached to membrane bound receptors, which further activates G protein. G protein sensitizes effector. Effector produces second messengers and second messenger causes cell response intracellularly. There are several different secondary messenger systems available, but they all are quite similar in overall mechanism, although the substances involved and overall effects can vary. In most cases, a ligand binds to a molecule to a membrane spanning receptor protein molecule. The binding of a ligand to the receptor causes a conformational changes in the receptor. These conformational changes can affect the activity of receptor and result in the production of active second messengers. The calcium ions are one type of second messengers and are responsible for many important physiological functions including muscle contraction, fertilization and neurotransmitter release.